All right, we'll keep moving here this afternoon. Our next speaker is Chong Lee. Chong is a postdoc at the University of Minnesota. He's done a lot of influenza research, and he's going to share with us today Influenza Reassortment, Survival of the Fittest. So welcome, Chong.
Thank you, everyone. Good afternoon. My name is Chong Lee from the University of Minnesota, and I'm very glad to share our ongoing research about the swine influenza reassortment happening in the field pigs. So a little bit background about swine influenza virus. Swine influenza virus infection is a costly disease in U.S. pig farms, and it happens throughout all production stages. Also, it's a genetic pathogen, so it can infect humans. So the prevent the swine influenza infections will benefit both for the swine industry and also for the human public health. So this kind of virus usually recognized by subtypes and genotypes. So based on their genome, because a genome made up by 8 single-strand RNA segments, and among them there are 2 segments responsible for coding the HA and A proteins, and they both determine the virus subtype based on which type of HA and A protein they are encoding. So for swine influenza virus, the most common subtypes are H1N1 and H3N2. So compared with the virus subtype, the influenza genotype are based on the whole set of 8 RNA segments. Based on the combination of the types of each of 8 RNA segments, the virus will further categorize into different genotypes. So benefit from the segmented genome, the 2 different influenza viruses could exchange their gene segments with each other and produce the progeny virus with a totally different gene combinations. So we call this process as a reassortment. And the newly generated progeny virus are also known as the reassortants. So compared with the parental viruses, the reassortants usually have a totally different genotype, but may still share the same subtype. So theoretically, 2 different parental viruses could produce up to 256 different genotypes of the progeny viruses. However, only a few of them could survive in the nature. That's because a successful reassortment process needs to meet multiple criteria. First, 2 different parental viruses need to co-infect within the same tissue of the same host. So after the virus propagations, they have a chance to meet with each other and exchange the gene segment within a single cell. So after the co-circulation at the cellular level, the virus genome from different, the gene segments from different viruses need to co-package together within a single variant. So at this stage, the incompatibility among the packaging signals and the functional proteins are the major constraining factors that inhibit the reassortment process. So the packaging signals located at both terminals of every gene segment, which is essential for the selective genome packaging of the whole set of influenza genome, and form the intermolecular network within a single variant. However, the packaging signals are varied by strains, so the incompatibility among the packaging signals always happens between the distantly related viruses, which bears them to produce the progeny virus with the specific genotypes. So the mismatch between the functional proteins is another very important factor, which will cause the fitness defect of the reassortants and makes them hard to survive in the nature. So when the reassortants were generated and released from the cells, they need to be selected from the environment. However, compared with the parental viruses, the majority of the reassortants are less fit, but they need to be compete with the parental viruses from the same environment. That's why only very few of the resultants are transmissible. However, the fitness is highly rely on the environmental conditions, and any factors like the host immunity, the drug treatment will shift the environment and change the selective preference for the resultants. So pigs are really good at result influenza viruses and also considered as a very important intermediate host for the influenza ecology. So that's because both human and avian-like influenza receptors are affluently co-expressed at their middle and lower respiratory tracts. And at the same time, the pig lungs provide a very suitable environment for the resultants to generate and survive, especially compared with nasal cavities. So that's why pigs were considered as a mixed vessel for the influenza virus, which kind of provide a platform for influenza viruses originating from different hosts to co-infect and resolve with each other. So as a result, at the farm level, co-circulation with different subtypes and genotypes are very commonly identified in the US pig farms, which in turn accelerates virus evolution and makes the virus unpredictable. So with varied genome sources, the high reassortment frequency and mutation rates, the influenza virus inherits the power of diversity. However, for the swine influenza vaccines, they usually need a long time to update. So the mismatch between the field and the vaccine strains presents a huge challenge for the vaccinations, and also facilitates the field strains to cause the breakthrough infections and circulate among the vaccinated pigs. So currently, there are several research, includes a study from our group that explores the influenza reassortment happen at the individual pig level. And all these studies have shown the individual pig can produce abundant number of resultants during the short period of time after the infections. And also our previous study shows the vaccination has the potential to reduce the emergence of the resultants by decreasing the duration of the influenza co-infections. However, all these studies are done under the experimental conditions. And based on our knowledge, there are no studies have explored the influenza reassortment that happen at the individual pig level in the field. So that's a major knowledge gap we address in this study. So to be more detailed, we more focus on how commonly are the resultants happen in the infected pigs in the field, and whether there are some difference between the resulted genotypes on their growing abilities on the cells from swine and human respiratory tract. So our objective is to identify the influenza resultants happening at the individual pig level under the field conditions, and evaluate their genotypic diversity and replicative fitness to better understand the emergence, the persistence, and subsidence of the novel influenza viruses. So this slide shows the overall workflow for this project. So first, we need to identify the pig farms that are experiencing the multi-subtype influenza co-circulations. So because that's a precondition for the influenza reassortment. And we collect the nasal swabs from the individual pigs during the 6-month surveillance. As each of the positive samples contain the virus populations made up by countless number of individual variants. So we perform the assay called the plague assay to separate these individual virus clones and cultures them individually. So we submit all the isolate influenza plaques for the whole genome sequencing. And after the bioinformatic analysis, we assign the genotype number to each plaque based on how different each of their 8 RNA segments with each other. And in the final, we test the growing ability of the selected plaques on the cells originated from the swine and human respiratory tract. So the nasal swab we use for this study are actually coming from our previous research that Nirmala published in 2020. Also, these nasal swabs are coming from the MAT study. So in the Nirmala study, we found the farm number 14 are experiencing the multi-subtype influenza co-circulations during the 6 months surveillance. And the virus circulating there has a very high genotypic diversity and also the high Shannon diversity at the pig population level. So there are 47 achieved nasal swabs from this farm that have already been successfully sequenced during the Nirmala study, and we use them all for the plaque assay. So among these 47 achieved nasal swabs, 23 of them are successfully performed the plaque assay, and we isolated 243 influenza plaques from these 23 samples. And all these influenza plaques are successfully whole genome sequenced by the Illumina platform. And after the row reads trimming and assembly, we construct the maximum likelihood tree for each gene. So all the reference we use for building the trees are actually coming from the OctoFlu, which is the automatic classification tools for the swine influenza virus. So we use a software called the Philopart for the cluster assignment. So this software will identify the subtree as a cluster if their median pairwise patristic distance is below a certain percentile threshold of the whole tree paristic distance distribution. So this is our standard for define a cluster. So for each cluster, they need to have at least 2 different samples and have at least 95% percentage of the ultra boost values. And also this chart shows the threshold we use to identify the cluster for each genes. So here is all the maximum likelihood tree we built for each gene. So within each tree, different colors represent different clusters. So we totally identified 23 clusters from 10 genes. So this is a summarized genotype table which shows the quantity of the influenza plaques by different gene combinations. And from the 243 influenza plaques, we found 212 of them are H3N2 virus. 9 of them are H3N1 virus, and 21 H1N1 virus, and 1 H1N2 virus. So all the H3 viruses belong to H3 C4A lineage, and the other H1 virus belong to H1 gamma lineage. So all the viruses could categorize into 24 different genotypes. And also about 6 out of 24 influenza genotypes were detected in multiple samples. So this bar plot shows the influenza genotypes identified in individual pigs. So each bar represents each pig, and different colors display the different genotypes. So from the 23 pigs in total, we found most of them are shedding the one kind of virus. But there are 8 pigs out of 23 have shed at least 2 different genotypes, have shed the influenza virus with at least 2 different genotypes at the same time. So among the 8 pigs, there are 2 pigs. There are 2 pigs shedding the influenza virus with a multiple subtypes. So you can see the pig number 40 they can shed as many as 10 different genotypes simultaneously, and which includes the H3N2 virus, H1N1 virus, and H1N2 virus. And the pig number 42 is shedding the H1N1 virus and the H3N2 virus at the same time. So from this colorful figure, so you can tell the rapid and extensive dynamics of the shedding virus from the individual pigs, especially when observed between the different months. Although this is still based on the limited number of pigs. So we also calculate the Shannon diversity index to measure the influenza genotypic diversity. So the left figure shows the influenza genotypic diversity at the individual pig level. So based on the number of influenza genotypes identified per pig. And the right figure shows at the farm level based on the influenza genotypes identified per month. So all the figures show the same trend, that is that the influenza genotypic diversity is continue decreasing during the 6 months surveillance time. So again, this is still based on the 23 pigs. So based on these results, we actually have more questions, which shows in the slides. So to answer these questions, actually, we need to test the replicative fitness of these virus plaques on the cell lines. So that's the first step. So how do we do it? So before I introduce the study plan, I need to tell the difference between the 2D and 3D cell cultures. So 2D cell cultures is normally we culture the cells, we seeding the cells on the cell culture plate and as the monolayers and merge them in the culture media. And for the 3D cultures, we actually see the cells on a special membrane and lift it up in the air. So all the cells will expose completely in the air, and they can suck the nutrients from the culture media underneath the membrane. So after the 3 to 4 weeks culture, so the cells will fully differentiate into a pristile epithelium. So the fully differentiated cells will produce the mucus and the cilius by itself. So it's a better model to simulate how the influenza virus replicate in animals' respiratory tract. So we use for the 2D culture, we use swine and human nasal epithelial cells to represent the upper respiratory tract and culture them at 33 centigrade. We also use swine and human bronchial primary epithelial cell to represent the middle respiratory tract and culture them at 37 centigrade. And we use A509, which is human lung epithelial cells. This is a passage cell line, so to represent both swine and human lower respiratory tract, and culture them at 39 centigrade. So for the 3D culture, we use swine trachea primary epithelial cells and human bronchial primary epithelial cells to represent the middle respiratory tract and culture them at 37 centigrade. So we inoculate the selected influenza isolates on each of these cell lines. So actually, we're still doing this part, and for this presentation, I just show part of the result on the human cell lines. So this is influenza replicative kinetics on the human bronchial epithelial cells. It's a 3D culture, so we inoculate them at the MOI 0.1. So as you can see, all the selected plaques could efficiently replicate on the human bronchial cells. You can see the virus titer rests up dramatically from the 3 to 24 hours after the inoculations. And we also, actually, we're more interested in if there is a significance on the virus titers between different genotypes of the plaques that isolate from the same host. So there are some significance show up, as you can see, labeled in red. So for the 2D cultures, here is a result of the influenza replicative kinetics on the human nasal primary epithelial cells and also the human lung epithelial cells, which culture at different temperatures. And compared with the 3D cultures, actually, there are more variations and more significant difference between the virus titers between the different genotypes of plaques, especially on the 48 and the 72 hours after the inoculations. However, the majority of the selected plaques could efficiently replicate on the cell lines derived from the human upper and lower respiratory tract. And also, we found some significant difference between different genotypes of the plaques that isolate from the same host during the sampling times. So the conclusion for this study, so first we found multiple distinct genotypes and subtypes of influenza virus can be shed simultaneously from the pigs under the field conditions. And also we found influenza strains obtained from the commercial pigs can replicate in various human epithelial cells from the respiratory tract, indicating the genetics potential. And also, especially for the virus tested on the monolayers, we found influenza virus with different genotypes showed displayed varied replicative fitness. And also, more efforts should be done on the swine farms, since pigs have a very high potential to produce diverse and genetic influenza viruses. And the efforts of the efforts to reduce the influenza genetic diversity should also be pursued. With that, I want to acknowledge my advisor, Dr. Monsi Termo and Marie Cohen, and also all my colleagues from the University of Minnesota. And also, I want to acknowledge Dr. Stacy and Victoria from the St. Jude Children's Hospital, as they provide us the swine cell lines and also the technical assistance, and also all the funding sources from the CVM program, signature program, and the USDA. That's all for my presentation. Thank you.
Thank you, Chong. Any questions from the audience? We have one right here.
Thank you for that presentation, Chong. Just a quick question. So the part of your study that you were looking at samples from the Nirmala study, where you saw, over the timeline, you saw the genetic diversity going down both within the pig and within the farm. Do you know if there was any correlation with the prevalence of positive pigs on the farm?
Sorry, we don't have those information, since actually those samples should collect at 2012 or 2013. So yeah, maybe we have information, but right now I have no ideas on that.
Yeah. Thank you.
Yeah.
John, very nice presentation. Do you have any thoughts about, since you found multiple isolates in these pigs, that there's selection going on, passaging it through human cells, or is there selection pressure? Is there quasi-species going on in these mixed populations of virus?
Actually, so we didn't do the whole genome sequencing on the virus after passaging on the different cell lines. Sorry, if I understand it right, your question is about if there is a connections between the replicated fitness with the prevalence in the farm. Is that right?
Or from the origins of the pigs' viruses on the farms, is there any pressure for different isolates to be selected?
Yeah,
including ones that could potentially grow in humans' cells.
Yeah, actually, the majority of these swine isolates from the plaques, swine influenza plaques, are affluently replicate on various human cell lines. So yeah, they have the ability to replicate on various locations based on the in vitro assay.
All right, we'll take Andres, then we have one over here too. Next. Go ahead. Yeah.
Okay. Great talk. I don't know if I missed it, but are all your isolates from nasal swabs, or did you took other samples? I'm thinking, did you consider perhaps taking tracheal swabs and see if the reassortments that are happening at different levels are the same or even euthanizing the pig? And have you measured some type of reassortments at different levels at the pig, different tissues?
That's a great question. Actually, in this study, all the samples are nasal swabs. But in our experimental studies, we use above the samples to isolate to see the influenza reassortment. Actually, there are some studies shows the above samples are better sample type to identify the influenza reassortment.
One over here.
Thanks, John. It was a very methodical, logical, lot of work. And so you say at the end we need to strengthen our surveillance. Are there molecular methods that can avoid having to do so much virus isolation, which Tom was saying might induce some bias? And how can, so is there unbiased molecular surveillance you can do that is a lot less work?
That's a great question. Actually, for my study, I just, because this is from the previous study, so actually they send, I remember 30 samples per month. So for my study, I just enroll all the samples in. But because for the plaque assay, it has a lower sensitivities, so only 23 of them are successfully enrolled, performed. Yeah.
I have one quick one before we get the next question here. Yeah. If I was reading the slide right there, you had a pig that had 10 or more genotypes in a pig. Is that correct?
Yeah, that's correct.
What are your just overall thoughts? Like what could potentially be leading to that pig having so many genotypes? Yeah,
actually, it actually occurs in many studies. Like there are few pigs super reassortant that they produce a super abundant number of reassortants. So actually, my guess is it's about the timing when you sample that pig. So yeah, that's my guess.
Sure. Congrats for your talk. When you do the hemagglutinin gene diversity inside of the same serotype, how is the cutoff that predict some antigenically escape between the strains belonging to the same subtype?
I mean, for the influenza subtypes, influenza subtypes are actually determined by the WHO, but it's not only based on the genetic similarities. It's also associated with the antigenic properties, whether different subtype could have the cross-reactivities. So it's more on the biological properties rather than the genetic relationship.
Thank you. we had time. That's probably
Yeah,
enough questions
thank you.
for you for now, Chong. Thank you very much. Let's give him one more round of applause.